Good evening, everyone. I'm great pleasure to welcome you all to the lectures of extraction of and isolation of phytohormones from plant tissues from past to present. My name is Andrzej Bajgus, and I am honored to be a part of this global initiative of academic network course. When we dive deep into the advances methodology determining plant hormones, particularly in the context of agriculture and biotechnology. Let's begin by looking at the fundamental role of phytohormones in plant biochemistry and plant biology. Before we dive into the specific of extraction and isolation technique, it's important to understand why phytohormones are and why phytohormones are so crucial in plant physiology. Phytohormones are naturally occurring organic compounds that influence various physiological processes in plants. They play an essential role in nearly every aspect of plant life, including growth, development, and responses to environmental stresses. This hormone acts as key signaling molecules, coordinating many biochemical processes, physiological and morphological processes too. The effect extends beyond individual cells. Phytohormones regulate nutrient allocation, determine the sourcing balance, and mediate plant responses to both abiotic and biotic stresses. Unlike in animals, where hormones are typically produced in glands, plant hormones are synthesized in various tissues and can function locally or be transported throughout the plant. The interaction between different phytohormones are highly complex, often involving synergistic or antagonistic relationships, then fine-tune plant responses. Understanding these plant hormones and their role is fundamental to developing effective methods for their extraction and quantification. Next, I would like to present that phytohormones exhibit a wide range of chemical structures. They can be broadly classified based on the biosynthetic origin. Most of them are derived from three key metabolic precursors. The first is amino acids, the second isoprenoids, and the third lipids. Amino acid-derived hormones include auxins, melatonin, ethylene, salicylic acid, and polyamines. Isoprenoid-based hormones there are such as cytokinins, brasinosteroids, gibberellins, abscisic acid, and strigolactones. Lipid-derived hormones such as yasmonic acid are synthesized from alpha-linoleic acid and are primarily, for example, involved in different responses and wound healing. Each of these hormone groups has unique biochemical properties and regulatory function. This diversity makes their extraction and quantification particularly challenging, as different methods must be tailored to the specific chemical nature of each phytohormone. With this understanding of the biosynthetic origins, we can now explore how specific metabolic pathways contribute to the production of different phytohormones in plants. They, of course, are formally distributed throughout the plants. They synthesize and transport it to specific tissue. This distribution depends on development stage, environmental conditions, internal signaling networks. For example, auxin I primarily synthesized in shoot apical meristem and young leaves. Cytokinin is produced in root tips and travel upward to promote, for example, cell division, shoot formation. Gibberellin are formed in growing tissues, such as young leaves and developing seeds. Ethylene is the smallest molecules, gaseous hormone produced in response to stresses. The next, for example, strigolactone are produced in roots and help regulate shoot branching. Understanding where these hormones are localized is critical for choosing the right extraction methods. Since the distribution varies across different plant organs and even with specific cell compartments, sampling extraction techniques must be carefully designed to target the precision location of each phytohormone. Over the years, scientists have identified 12 major classes of plant hormones, each with a distinct chemical structure and biological function. This includes auxin, cytokinins, brasinosteroids, salicylic acid, and other 2 peptide hormones. Each phytohormone is structurally unique, ranging from simple molecules like ethylene to complex polycyclic structures as gibberellins and brasinosteroids. This diversity in structure and function means that the single extraction methods cannot be universally applied to all phytohormones. Instead, customized strategies must be used depending on the hormone's chemical nature, polarity, and cellular localization. We know that phytohormones can be categorized based on their chemical nature and solubility. Auxin and melatonin are indole derivatives, making them relatively hydrophobic and requiring organic solvents for extractions. The next phytohormone, abscisic acid, belong to the sesquiterpene family, while gibberellins are tetracyclic diterpenoid acids, both of which have moderate polarity and required mix of organic and aqueous solvents for efficient extraction. On the other hand, cytokinins are adenine analogs, meaning they are relatively polar and regulate aqueous-based extraction methods. Brasinosteroids are polyhydroxysteroids, making them structurally similar to animal steroids. For example, estradiol, estrogen and androgens, sex hormones, and requiring lipid-based extraction methods. Yasmonates as fatty acid derivatives, making them hydrophobic and requiring organic extraction approaches. Ethylene, as I said, there's simple gaseous molecules, presents unique challenges for extraction, requiring tapping techniques rather than conventional liquid phase methods. Strigolactone are terpenoid lactone, making them slightly hydrophobic and requiring careful handling during extraction. Generally, auxin, gibberellin, salicylic, and yasmonic acid are acidic, cytokinin alkaline phytohormones. Thus, the diversity of chemical structures and properties of different phytohormones make the specific extraction, purification, and quantification, as well localization procedures necessary. This slide shows different structures of phytohormones. What about history? In the early stage of phytohormone research, bioassay, biotest were commonly used to estimate hormone levels based on the physiological effects on plants. This excluded methods like, for example, the Avena test for auxin or seed germination bioassay for gibberellin. Why bioassay provides some insight into the presence of hormone. They lack precision and specificity, making them unreliable for detailed quantitative analysis. Later, immunoassays such as ELISA introduced a more quantitative approach by using antibodies to detect specific hormones. However, these methods were still limited by the specificity of the antibodies used, leading to potential cross-reactivity and errors in hormone quantification. A major breakout in plant hormone analysis came with development of chromatographic techniques. High-performance liquid chromatography, HPLC, became a widely used tool, allowing scientists to separate and quantify multiple phytohormones with much greater accuracy. The next techniques, gas chromatography, particularly coupled with mass spectrometry, provide even better sensitivity and specificity, especially for volatile hormones such as ethylene. Over one of the main limits of GCMS is that the certain hormones required a derivatization step to make them volatile enough for analysis. The extra step increases processes time and introduces potential sources of errors. Over the past few decades, the field of phytohormone analysis has undergone significant advancement. Different analytical techniques have been developed, each building the previous method to improve sensitivity, accuracy, and efficiency in hormone detection. In 1980s, GCMS became powerful for analyzing volatile phytohormones. The next method popular in 2000 was LCMS techniques. Unlike GC, gas chromatography, liquid chromatography does not require derivatization, making it a faster and more versatile options for analysis different classes of phytohormones. Introduction of tandem mass spectrometry further enhanced the accuracy of phytohormones detection. These techniques allow to greater sensitivity, enabling researchers to detect more hormone at extremely low concentrations, sometimes down to the picomolar range. It also improved specificity by distinguishing between similar molecules, ensuring that plant hormones and their metabolites could be measured. Accuracy. One of the major challenges in phytohormone analysis arise from the complex of plant extract. Unlike simple chemical solutions, plant tissue contain a vast array of biological molecules, for example, pigments or carbohydrates. These substances can interfere with the detection and quantification of phytohormones, making a specific extraction and purification method necessary. A key difficulty in phytohormone analysis is that endogenous phytohormones are present at trace levels, often in the range nanogram to picogram per gram fresh weight. However, these hormones exist in mixture with the hundred of other plant compounds. This is a problem to resolve. Another challenge is the chemical diversity of phytohormones, like auxin and gibberellins are acidic, whereas cytokinin are alkaline. Some hormones are hydrophilic and dissolve easily in water, while others are lipophilic and require organic solvents for effective extraction. This diversity makes it impossible to use a single universal method for all phytohormones, requiring multiple approaches tailored to the specific chemical properties of each phytohormone. In this slide, I present the graph on which concentration and the properties of plant hormones and secondary metabolites in plant. There are maybe two interpretations. The first, cytokinin and gibberellins have moderate polarity and concentration generally between from 1 to 10 nanogram per gram fresh weight. Auxin and abscisic acid, yasmonic acid, salicylic acid have lower polarity than cytokinins, but higher concentration ranging from 10 to 100 nanogram per gram. On the other hand, strigolactons and brasinosteroids are the least popular polar hormones and are found at much lower concentration from 0.1 to 1 nanogram per gram fresh weight. Secondary metabolites have significant higher concentration and cover a broad range of polarities. The second interpretation is that since strigolacton and brasinosteroids are present at very low concentration, highly scientific analytical techniques, for example LC-MS, are essential for the quantification. What is the conclusion for challenges in phytohormone isolation? The first, low concentration in plant tissues. Required pre-concentrated steps, for example, solid phase extraction, complexity of plant matrix, co-extraction of interfering substances, for example, pigments, phenolics, sugars, and difficulty in separating hormones from other metabolites. What is the approach to solving these problems? The first, employing selective extraction methods, and the second, utilizing advanced chromatography for separation. Some preparation is of course, is one of the most critical stages in phytohormone analysis, as it directly influences the accuracy and reliability of these results. This process involves breaking down the plant matrix, isolating the target hormones, and eliminating interfering substances to ensure precise quantification. The whole analysis procedure of phytohormones based on LCMS recommended for typical plant materials can be divided into 5 stages. The first is sampling, the second grinding, the third extraction, the fourth purification, and the last detection. Proper sample preparation typically accounts for 70–80% of total analysis time. It's important. Why? Highlighting its significant ensuring reproducible results. It keys steps such as homogenization, extraction, purification, and sometimes derivatization. Sometimes derivatization. Depending on the hormone type and the analytical method used. The main goal of sample preparation is to decompose the plant matrix, making the phytohormones accessible while preventing degradation. Given the complexity of plant tissues, techniques like solid phase extraction or liquid extraction are commonly used to concentrate and purify the target hormones before final detection. The accuracy of phytohormone analysis begins with proper sample collection and storage. It is important since plant hormones are highly dynamic and can rapidly degrade or change in concentration due to the environmental factors. Carefully handling is essential to maintain these original levels. When collecting plant tissue samples, rapid manipulation with minimal disturbance is crucial. Even minor stress, such as cutting, can trigger hormonal changes within seconds. Therefore, sample must be collected quickly and processed under controlled conditions to prevent alteration in hormone levels. After harvesting, plant material are typically flash frozen in liquid nitrogen to stop enzymatic activity and prevented hormone degradation. The samples are then stored at -80 Celsius degree until processing. For certain phytohormone storage conditions must be optimized further. For example, some hormones degrade in light and require dark storage, while others are sensitive to oxidation. In order to calculate plant hormones concentration in plants accurately, the weight of plant materials need to be recorded exactly. In different methods of quantification, for example, auxin or cytokinin, abscisic acid, both dry weight or fresh weight are used to express plant hormone concentration in plant tissues. For fresh weight values, frozen fresh tissue can be weighted, while for dry weight values, the tissue can be lyophilized and then weighted. Once plant samples have been properly collected and stored, the next important step is homogenization. This process involves breaking down the plant tissue to release phytohormone, making them accessible for extraction. Proper homogenization ensures that hormones are uniquely distributed within the sample and improves extraction efficiency. To prevent degradation, homogenization is often performed under liquid nitrogen. Sometimes we use for larger plant sample homogenization using a mortar pestle, but sometimes special homogenizators. For small amounts of tissue, micro-homogenization techniques such as vibrating ball micromills is used. Success of any phytohormone extraction process depends on efficient homogenization. Once the plant tissue has been fully ground and cell walls broken, the next step is extraction of phytohormone into a suitable solvent, ensuring optimal recovery for the next analysis of the next detection of plant hormones. The extraction is essential for isolation target hormones with complex plant matrix and transferring them into a liquid phase for next purification and detection. Phytohormone extraction is challenging due to the low concentration of these compounds and the presence of numerous interfering substances such as what I said, pigments, proteins or sugars and other structural components. An ideal extraction method must be selective, efficient and capable of preserving hormone stability throughout the process. One of the most widely used strategies is solvent extraction, where a carefully chosen solvent dissolves the phytohormones while minimizing the extraction of unwanted compounds. The choice of solvent depends on the chemical properties on the target hormones. I said on the beginning lecture about it. Hydrophilic hormones like cytokinins are best extracted using aqueous solvents, while lipophilic hormones such as yasmonate or abscisic acid required organic solvent like methanol, acetone or chloroform. In many cases, a mixture of solvent is used to optimize extraction efficiency and improve hormone recovery. Depending on different extraction modes, this method can be divided into classic and micro extraction methods. Both classic extraction and micro extraction methods are based on liquid or solid phase extraction. Classic extraction methods provide no complete extraction efficiency. Compared with classical extraction methods, micro extraction have advantages of having high extraction efficiency and miniaturization of device enrichment capacity, making them more suitable for the analysis of phytohormones. The extraction and cleanup techniques for trace compounds analysis include liquid extraction, liquid phase extraction, micro extraction, dispersive liquid micro extraction, polymer monolith micro extraction, and etc. etc. Compared to the traditional liquid extraction methods in which analytes are divided between different solvents, solid phase extraction is becoming the preferred purification techniques used in phytohormone quantification for its advantages, saving time and solvent and its higher recovery and throughput. We know that solid phase extraction is one of the most effective techniques for purifying phytohormones before analyzing. This method allows for the selective isolation of target hormones while removing interfering compounds. SPE solid phase extraction works by passing the extracted sample through the cartridge filled with a specific sorbent material. Depending on the chemical properties of the phytohormones, different types of cartridges are used. For example, C18 for nanopolar hormones, or anion cartridges for acidic hormone. Cationic and anionic exchange cartridges are used. The retained hormones are then eluted with an appropriate solution, resulting in a purified extract ready to detection. SPE offers several advantages over traditional liquid-liquid extraction, including higher recovery rates, reduced solvent use, and improved reproducibility. This technique is now a standard step in phytohormone quantification, ensuring more precise and reliable results in phytohormone research. Along the organic solvent tested, 80% methanol has become a preferred solvent for relatively good extraction efficiency for the most phytohormones. This table shows a comparison of solvent efficiency, which is easy to interpret. In addition to a single solvent, mixtures comprising different solvents has been frequently used. For example, the modified Bielewski's solvent, methanol forming acid, water in range 15 to 1 to 4 showed better extraction efficiency than a single solvent for simultaneous extraction of multiple phytohormone categories with inhibition of enzymatic degradation. The next step in this procedure is ultrasonification, which is a powerful technique used to enhance the efficiency of phytohormone extraction. This method applies high-frequency sound wave to break plant cell walls, allowing for better release of intracellular hormones into the extraction solvent. One of the main advantages of ultrasonification is its ability to reduce extraction time while improving hormone recovery. It also enhances mass transfer, making it particularly effective for extraction hormones for tough or fibrous plant tissue. Additionally, ultrasonification minimizes the need for harsh solvent and high temperature, which helps preserve the integrity of high sensitive phytohormones. Because of these benefits, this technique's ultrasonification is widely used in modern phytohormone analysis to improve both efficiency and accuracy. Of course, a few types of ultrasonification exist. For example, ultrasonification assisted extraction, microwave assisted extraction, vacuum microwave assisted extraction, and other. In addition to conventional solvent extraction methods, advanced techniques such as microwave assist extraction or vacuum have been developed to improve the efficiency of phytohormone isolation. Both methods offer faster, more efficient and environmentally friendly alternative to traditional extraction methods. These techniques are particularly used when dealing with complex plant matrix or when working with delicate, sensitive phytohormones then require gentle handling during extraction. Liquid-to-liquid extraction is, of course, used method for isolation phytohormones from plants. This technique is based on a principle that solvent can dissolve itself between two solvents, typically aqueous or organic phases. By carefully selecting this solvent, phytohormones can be efficiently separated from plant components. This extraction, this method, is particularly used for removing unwanted polar or non-polar impurities. Hydrophilic phytohormones such as cytokinins are extracted into aqueous phase, while lipophilic into organic phase. Now, some improved liquid-to-liquid extraction methods are developed to simplify sample preparation procedure, including single drop microextraction, wetting film extraction, homogeneous liquid-to-liquid extraction, and other. Solid phase extraction using cartridges filled with matrix compounds, which extract compounds based on diverse interactions, including adsorption, hydrogen bonding, polar and non-polar interactions, cation or anion exchange, or size exclusion, have been widely used for high throughput phytohormone extraction and purification. Many SPE columns, such as SEPAC C18, Waters Oasis HLB, Oasis MCX, and Oasis Max, containing different sorbents, are now commercially available for phytohormone purification from crude extract, so that SPE has become the mainstream method for purification of phytohormones. I like this column, I like these methods, of course. Mixed mode, reverse phase cation exchange, anion cation cartridges are commonly used to purify acidic plant hormones like brasinosteroids, leveraging the two types of interactions to enhance the sensitivity of LCMS-MS quantification. Another type purification methods employing C18 and mixed mode sorbents are also utilized to quantify multiple phytohormones simultaneously, for example, auxin, yasmonic, salicylic, abscisic acids, gibberellins, cytokinins, brasinosteroids. Another type of extraction method is solid phase micro-extraction, which is more recently developed for SPE than use fibers coated with an extracted phase, which can be either a liquid, polymer, or liquid sorbent. This technique enables the extraction of volatile and non-volatile analytes from various media, including liquid and gases. SPME offers the advantage of high recovery rate, greater than 80%, combined with simple and efficient operation in extracting and purifying phytohormones. The next extraction technique is a magnetic solid extraction. Is and improve SPE for enrichment and purification of target analytes from large volumes based on the use of magnetic. In this procedure, a magnetic sorbent is added to a solution or suspension containing the target analyte. The analyte would be adsorbed onto the magnetic adsorbent, and then the adsorbent is adsorbed, analyte is recovered from the suspension using a magnetic separator. The slides show this this procedure, and after extraction, the next essential step in phytohormone analysis is purification. Since plant extract contain a complex mixture of compounds, purification is necessary to remove interfering substances that could affect the accuracy of hormone quantification. One of the most commonly used purification methods is chromatographic separation, including thin layer chromatography, TLC, immunoaffinity chromatography, or high-performance liquid chromatography, HPLC. These techniques allow for the selective isolation of phytohormone based on their chemical properties. Purification not only enhances detection sensitivity, but also improves the stability and reproducibility of this analysis. Without a proper purification, contaminants in plant extract could interfere with mass spectrometry or chromatographic separation, leading in inaccurate results. Once purification is complete, the final step in the workflow is detection and quantification, which provides precise measurements of phytohormone concentration in plant tissue. Purification and cleaning crude extract from plant sample can remove the bulk of interfering compounds and improve separation by HPLC and detection by MS. The choice of purification methods depends on not only the target analytes, but also on the type of analysis to be performed, as well as it's very important for us, the available analytical instruments. In addition to these methods, such as quick, easy, cheap, effective, large, and safe catchers, and dual cloud point extraction are also explored in phytohormonal extraction and purification. What is important sometimes to use is derivatization. This process involves chemically modify the hormone molecules to enhance their stability, detectability, and volatility, making them more suitable for analysis. Derivatization is particularly important in GCMS, where certain phytohormones such as auxin, abscisic acid need to be converted in more volatile forms. Common derivatization techniques include methylation and silylation, which improve the separation and sensitivity of these compounds during analysis. The stabilizing the target hormones and improving the chemical properties, the derivatization ensures that even low concentration phytohormones can be detected with high precision. This step is essential in case where direct detection methods do not provide adequate sensitivity or selectivity. We must remember about GCMS, or when we detected phytohormones using by LC-Q-TOF MS. This slide is the last slide in my lecture. The final step, detection, I would like to present. In the lecture hormone profiling in plant tissue. Thank you very much for your attention. You must easy. The phytohormone detection is very easy, but we must have a very good procedure to isolation. From extraction, purification, and other steps must be complete. If you have any questions, I suggest write me on my email, and I try to very quickly answer on the questions. If you Thank you very much. Thank you.
Thank you, Professor Andres, for your nice lecture. So, the next lecture will start at 3:30, and that is basically a hands-on training on water potentiometer, and it will be by Dr. Yamshi Arif. Okay, just wait for a few minutes, then we have a hands-on training inside the lecture theater on the water potential.